Hi, Ryland. Thanks for joining me today. I appreciate it.
No worries. Thanks for having me.
Absolutely. So I'm sure you get asked this question a lot. Can you introduce the concept of FMT to an unfamiliar audience?
Yeah, for sure. So fecal microbiota transplantation, or FMT, is um, essentially a transplantation of living bacteria from a donor that has a healthy gut microbiome, and is transplanted into a recipient. These have been researched quite extensively, especially recently, and they're growing in popularity for treating C. diff recurrent infections. So these are in patients that have had a major GI surgery and they have a recurring bacterial infection in their intestines that is not being resolved with antibiotics. These patients are reporting really low quality of life. They have diarrhea that will not resolve. It really is just awful for the patients, and often providing antibiotics does not fully treat or resolve these recurrent infections. So the FMTs are then given to patients in order to change what bacteria are present in their gut microbiome and are inhabiting their, um, living in their gut, and these then resolve the recurrent C. diff infection and contribute to improving their quality of life. These have also been kind of researched for now for treating different diseases as well, both GI diseases and also even some researchers explore mental health and the links that has, is, that the gut is linked to mental health as well as overall physical health and our well-being. So these are very important for not only just recurrent infections, but might be important for just improving the quality of life of numerous patients who are affected by multiple illnesses.
Absolutely. Before we kicked off the discussion, you had mentioned that this was a very large study, piece of research that you participated in. So there were certainly a number of different analytical platforms chosen. And luckily for us, this podcast, this interview series focuses on chromatography mass spec. And you noted that GC-TOFMS was one of the approaches. So I was hoping you could talk to us about how that platform was used and the unique insight that it provided.
Yeah. So I'm by training a chromatographer, and my research interests are in metabolomics and multidimensional chromatography. My PhD, which is where this research was done, was in comprehensive two-dimensional gas chromatography, so GC-TOFMS, MS for analyzing complex matrices that are often for metabolomic studies. So the sort of motivation for using GC-TOFMS GCMS for this was because bacteria in fecal microbiome transplants and in stool as well, they produce a lot of volatile compounds that are biologically relevant. These include short chain fatty acids. So the short chain fatty acids are produced by bacteria. They're quite volatile, so they're difficult to detect for not using gas chromatography, and they also have been linked to gut health as well. So they seem to be important in mediating gut health and potentially interacting and having a positive effect on gut health. So I developed the method using solid phase microextraction, SPME, and just straight from the FMT cultures, you're able to detect on the short chain fatty acids and other volatiles as well without having to derivatize them. So not changing um, not actually going in and altering them chemically, just detecting them just by heating up the sample, forming them into a fiber, and then putting them onto the column. I shared two papers with you, and with the other paper as well, there was looking at polar metabolites too. So trying to look at polar, slightly larger, small molecules as well. So this is achieved again by using GC-TOFMS GCMS to look at these polar metabolites for derivatizing, adding a TMS group onto the metabolites before analyzing. And I think GC-TOFMS GCMS can play in multidimensional separations will play a really large role in metabolomics and in metabolite profiling going forward in the future because of how many metabolites you can see in a single separation. So we can routinely detect thousands of individual analytes in a single GC-TOFMS GC separation. This is incredibly powerful for these sorts of untargeted studies where, okay, we know short chain fatty acids are important, but what other short chain or small volatile molecules are present that may be contributing positively or negatively to the effect that we're trying to observe. So I think that this is a very important analytical platform, and going forward will be used more in sort of clinical research, where researchers seem to be able to detect as many small molecules as possible.
It's been really interesting for me to learn more about GC by GC, and part of it's through this interview series, but also Organimation, my company, just has more people using our evaporators to concentrate samples ahead of that analysis. I was hoping you could walk us through any sample prep challenges you faced, and there's certainly the differences between the live versus the dead bacterial fractions, I think would be interesting to discuss.
Yeah, so of course, one challenge is dealing with biological samples and getting them into a format where we can analyze them by GC. So we can't just take a live bacterial fraction, might be in water, inject it straight onto a GC, it'll destroy the instrument. So I was worried about trying a derivatized method for switching fatty acid. So instead, that is why just looking at the FMT culture fractions that were in aqueous solution, we just added salt to them and then stuck an SPME fiber in the headspace, heated them up a little bit. I was able to absorb all these volatiles on relatively quickly within a few minutes of the SPME fiber. So these were, with this is minimal sample preparation, which is good for analysis. And also it's not destructive. So we're not potentially losing short-chain fatty acids and introducing two false negatives into the analysis. So that was really powerful for looking at the polar metabolome. It's also greener too. So the only reagent used was sodium chloride versus some other derivatization reagents that could be quite environmentally toxic. Looking at the polar metabolites, it was that is trickier. Fecal samples, the solid samples, you have to get into the liquid state before you can derivatize and analyze it. The other fecal samples were already, or the other FMT samples already liquid, which made it a little bit easier to work with. We still had to extract the methanol and then dry them down and derivatize to dry them down using one of your devices. I think that one's quite old. It's well not old enough to it be in the competition that I've seen advertised on LinkedIn, but had to dry them down and then derivatize them. Of course, keep water out of the samples. One benefit of GC times GC as well is you resolve a lot of the analytes away from the derivatization reagents. So when you add derivatization, you got a huge band of TMS, siloxane compounds that are streaking across the chromatogram, but your analytes are dissolved from that in the second dimension. So you can achieve lower limits of detection, more pure mass spectra, which is important when you're doing a complicated sample preparation like this versus a 1D GCMS, where the background is going to be all these derivatization reagents, so it makes it difficult to see low abundance molecules that might be clinically relevant.
Well, I'm always happy to hear about a happy user of our equipment, certainly, but also the idea that just in general that there were many challenges with your sample prep and that it seemed like a pretty streamlined process, which is great to hear.
Yeah.
What was the most difficult aspect of measuring short chain fatty acid production in vitro, and how were you able to help overcome that with the GC by GC?
So one big challenge is they are volatile, of course. So we put them on the instrument and trying to make the analysis go as quick as possible. So samples are sort of prepared on the fly rather than preparing a whole batch of 100 samples and then racking them on the instrument and leaving them sit overnight. So that was the biggest challenge. And that just meant I had an undergrad technician working with me, and we were just preparing samples kind of on the fly, preparing one or two samples as we go. Another difficulty as well is on the quality control side. When you're analyzing samples on the fly like this, you can't just go and make a pooled sample of all the samples you're analyzing, because then you'd have to say if they came to us frozen, so you have to thaw the samples and then refreeze them. So the freeze-thaw cycle is just an important consideration when looking at biological samples. So those were both important considerations, and wouldn't necessarily say that GC times GC made it any easier, but having a really reproducible method with SPME that didn't involve complex sample cleanup for looking at short-chain fatty acid made say, getting the kind of doing the sample preparation on the fly a lot more accessible.
You found that bacterial viability was fairly stable across frozen formulations, but that storage duration reduced the diversity, let's say. From a therapeutic perspective, how important is this finding for defining shelf life guidelines?
Yeah, so with any live biological sample or treatment being given, one challenge to overcome is these are live cultures. They change with time. You have to preserve them as well to keep them alive so they still have efficacy. It's not like a small molecule compound that is in a pharmaceutical in a pill that's shelf stable at room temperature for 10 years. Unfortunately, there are other considerations. Clinically, there's a trade-off for the convenience that comes with a reduced shelf life. And also that we need to monitor these FMT products closely to ensure that they're not degrading over time. And this is something I think that the role of GC-TOFMS actually to play an important part is you could use, say, analytical technique like this to monitor chemical cues to determine if these products are degrading or not, and then correlate this back with clinical efficacy in future studies. So I think that there is linking it back to the analytical side, which is where my interests are. I think there is, it has, of course, analytical chemistry plays an important role in our day-to-day life, but I think this is a particularly important part of findings on the analytical side is you can use it to gauge the efficacy and monitor FMT products before clinical use.
Totally. Now, I know this is a pretty broad question, but I'm sure you have some thoughts on it. Purely from that analytical chemistry standpoint, what tools or methods do you think will be most important for next generation microbiome therapeutics?
Yeah, so of course I have a bit of bias here. I think that chromatography coupled to mass spec is going to be the most important. If we're looking at small molecules, it not only are small molecules produced by bacteria and they have beneficial, you know, uh, they interact beneficially with human health. So potential that certain additives may also change formulations of FMT products. We can use them, as I mentioned in the previous session, as well to monitor how these products are they shelf stable? Are they degrading? On the fly, QA, QC, or QC to all these products before they're being used in the therapeutic setting. Of course, though, it's not just one analytical method that's going to be important going forward. So 6ES sequencing, perhaps are important questions that people that are interested in proteomics may have. I think that these are all going to be a team effort across all fields of analytical and bioanalytical sciences to work with these living biological samples.
Always a team effort for sure. How might findings from your research inform the design of synthetic microbial consortia?
Yeah, so at least on the side of the GC-TOFMS GC, we know that short-chain fatty acids are important. So perhaps one obvious use of these results in designing sort of synthetic or defined therapeutics would be trying to mirror what the metabolome looks like, both the volatile metabolome and the non-volatile metabolome. So what does the global metabolome look like to ensure that we are preserving what small molecules are actually present in these samples, since we give these to patients, so not only ensuring that are those the beneficial microbes there, but what small molecules are there. Are the bacteria producing these relative small molecules such as short-chain fatty acids, so that these products continue to have high clinical efficacy and therapeutic use. So that's where I think that the findings of this research should help. So going forward, as you mentioned, with synthetic microbial product, is trying to replicate the metabolome.
You just touched on the importance of chromatography mass spec moving forward. But just in general terms, what excites you about microbiome science in the coming years in terms of progress that'll be made?
I would say I think just all the unique applications that researchers continue to find for our technologies. I'm really on the technology development and the data analysis side. I do have biological questions that interest me, but I'm not particularly, my research program isn't looking at one pathway or one bacterium. I'm always just excited where I see some cool new problem that involves microbes or other biological samples. Someone has taken GCMS or GC times GCMS and gone and analyzed really cool sample. At least in my own research group, since I started as a professor at the University of Victoria in Canada a couple months ago, comprehensive two-dimensional liquid chromatography, mass spectrometry for metabolomics. And this is an area that really excites me for metabolomics because right now with one-dimensional LCMS, you get a lot of powerful results. We can see lots of biologically relevant compounds, but I suspect that a lot of the, similar to how GC-GC has become very important and powerful, I suspect that we'll be able to cover more of the metabolome in a single analysis with LC times LCMS. Also be able to get more valuable insight from less samples as well. So we'll be able to do one analysis with an LC times LCMS. And just that, sure, we can't do statistics on this, but actually go and look at what compounds are present and then make some biological decisions or discover new compounds or new metabolites that might be clinically relevant, that might inspire new drugs, pharmaceuticals, and then use these in future studies. So these are the areas that get me really excited. Of course, like I said, I have a bit of bias. I'm really excited about the comprehensive multidimensional separations, both GC times GC and LC times LC. I just like seeing the application. So yeah, that's where my excitement lies. And I'm always looking for collaborators as well that are looking to study and solve some unique problems with small molecules. And I'm trying to get a LC times LCMS system in the next year.
That's great to hear.
Yeah.
I should have mentioned earlier to you that the audience for this interview series is undergraduate researchers.
Okay.
For people in that spot just beginning their career, what else should they know about this research from your perspective?
Yeah, so for undergrad, I would say the most important thing to be aware of is the data processing and data handling that's required to work with metabolomics data. If you're really interested in metabolomics, you have to have a really strong data analysis skill set. So that includes understanding statistics, understanding how to code, being able to use Excel proficiently. These are all really important skills. And I think in a lot of undergraduate chemistry degrees, skills that are often not taught to undergrads. So they either have to, say, do a minor in computer science or learn these fields on their own. But when a lot of people think of analytical chemistry, they think of just measuring one or two molecules and maybe doing a t-test and then that's it. But in targeted metabolomics and compound discovery, there's a lot of multivariate statistics that's required. So having a really strong background in data analysis is a really important skill that complements all of the other technical skills that you learn in a laboratory, in an analytical lab during an undergrad degree. Knowing data science never hurt. And I think that pairing a strong background in, say, computer science or data science with another one of the physical sciences, like the analytical chemistry or organic chemistry or physics or biology, creates and opens a lot of doors and creates a lot of opportunities for undergraduate researchers going into graduate school. I know in my research group, I'm always looking for people that are able to code and understand data and do statistics, because there's a lot of really powerful insights we can obtain from this that if you don't have the skill sets, it's a lot more challenging to go in a process and analyze complex untargeted data.
Every analytical chemist I've spoken with has been very quick to point to the fact that to really excel in the field, you do need skills from different areas. And it's not just the chemistry focus. It could be the math or the physics, and certainly the data work, as you mentioned, is critically important. I think that's a great thing for undergraduate students to keep in mind, that it's really how well you can bring together your experience and skills from different areas and combine them to help your chemistry research.
Yeah, and I also see now a lot of discussion around concern that a computer science degree is employable as it was 5 years ago or 10 years ago. And well, I think that's true. I think having other skills that complement computer science still make people employable. So it's important to have other skills that complement this as well, because people aren't just coding for the sake of coding. They are using computer science and coding to make software that enable more exciting future research, that enable discoveries in research, and also enabled partners and people in industry to achieve their goals as well.
Just my 2 cents, but I certainly think that the creation of AI, the concept that it has destroyed the need for knowing coding, is pretty exaggerated.
Yes, I agree. It's still important to, while AI can write code, if you don't understand how, you should understand how to implement that, or even how to execute a line of code. There's nothing you can do, right? It's not, sure, AI, ChatGPT can write code, but if you don't understand how coding works or the types of problems the coding is really good at solving, then I don't think it's that useful. So that's why I say having a really strong background that complements this. You can use, say, these AI tools to help write code faster, but you can't necessarily excuse yourself from learning data science altogether.
I totally agree. Ryland, thanks so much for joining me today. I really appreciate you sharing with the audience, and hopefully at some point in the future we could do a follow-up to see how things have progressed.
For sure, that's great. Thank you so much for having me today. I really enjoyed our conversation.